right, let's talk about the body's tiny engines, enzymes. These things are the absolute unsung heroes, right? They're working nonstop inside every single one of your cells, and honestly, they're the reason life itself is even possible.
I mean, have you ever really stopped to think about it? Right now, as you're listening to this, you're digesting food, your muscles are making tiny adjustments, you're even thinking. All of that, it's just trillions of chemical reactions happening every single second. So how does your body pull this off perfectly without, bursting into flames? Well, the secret, it's all about those tiny engines.
100 million. That number is it's pure power. What it means is that enzymes can make a reaction happen up to 100 million times faster than it would on its own. To really get what that means, think about your lunch. Thanks to enzymes, you'll digest it in a few hours. Without them? Yeah, we'd be talking centuries. No joke.
And I'm not exaggerating here, not even a little bit. Without these biological catalysts, all the reactions that keep us alive would grind to a halt. They'd be so impossibly slow. You can think of them as the gatekeepers of the speed of life. Bottom line, without enzymes, we just wouldn't exist.
Okay, so let's dive into our first section. The best way to picture an enzyme is to think of it like a tiny, microscopic machinist. Seriously, each one has a super specific job on this huge, complicated assembly line that we call metabolism. So let's get to know these machinists a little better.
So here's the textbook definition. An enzyme is a protein that acts as a biological catalyst, speeding up a reaction without being consumed. Let's unpack that. First off, they're made of protein. Super important. Second, they're catalysts. Fancy word for something that speeds things up. And third, and this is the magic part, they're reusable. They do their job and then they're ready to go again over and over without getting worn out.
So what really makes these little machinists tick? Well, for one, they are incredibly specific. I mean, one enzyme usually does one and only one job. And how do they do that? They have this special little pocket called the active site. Think of it like a perfectly shaped keyhole that only one specific key or molecule can fit into. And just like any delicate machine, they're kind of fussy about their work environment. They're really sensitive to things like heat.
So, what does this process actually look like in action? It's this elegant, super fast, four-step cycle. First, the enzyme finds its target molecule, we call that a substrate. They lock together, then, bam, the chemical reaction happens. After that, the enzyme releases the new finished product. And here's the kicker. It's immediately free to do the whole thing again and again, thousands of times a second. It's just mind-bogglingly efficient.
All right, section 2. So if our bodies are like a giant workshop, you can't have just one type of machinist, right? You need welders, cutters, builders, the whole crew. Well, it's the exact same with enzymes. They have a whole playbook of different jobs. Let's meet the team.
First up, you've got the breakers. The sciencey name is hydrolysis, but their job is basically demolition. A great example is an enzyme called amylase. It's right in your saliva. The second you take a bite of bread or pasta, amylase is on the job, breaking that complex starch down into simple sugars you can actually use. Then we have the remodelers, or oxidoreductases. These guys aren't about demolition, they're about renovation. They move electrons around, changing one molecule into another. And you've definitely felt these guys at work. You know that burn in your muscles when you're going hard in a workout? That's lactate dehydrogenase, a remodeler enzyme cleaning up the byproducts of all that energy you're burning.
Meet the group swappers, or transferases. I like to think of these as the molecular-level movers. Their entire job is to pick up a functional piece from one molecule and stick it onto another. It sounds simple, but this is absolutely fundamental for building pretty much everything important, like new proteins.
And last but not least, we have the builders, the ligases. Their role is pure construction. They take two separate things and stick them together. But construction takes energy, right? So they use a little bit of power from a molecule called ATP. You can think of ATP as the body's rechargeable battery, its main energy currency. It provides the juice these enzymes need to build bigger, more complex stuff for us.
Okay, on to our third section. So we've established these machinists are amazing and powerful. But they're also kind of high maintenance. They're very particular. Their workshop conditions have to be just right, or they just won't work properly.
Let's start with temperature. As you can see from this chart, our enzymes have a happy place, a real sweet spot, right around normal body temp. If you get too cold, they just get sluggish and slow down. But if you get too hot, think about a high fever, that's when things get dangerous. They literally start to unravel. They lose their shape. It's a process called denaturing. And once that happens, they're broken. They just stop working.
And it's not just temperature. pH balance is also a huge deal. A perfect example is pepsin. This is the enzyme in your stomach that breaks down protein, and it loves acid. I mean, it works best at a pH of 2, which is incredibly acidic. But if you take that same enzyme and drop it into a non-acidic or alkaline solution, it gets totally wrecked. It just falls apart.
All right, section 4. So it's pretty clear that when these little machinists are all happy and working correctly, we're healthy. But what happens when they're not? Well, that can be a major sign of trouble, and this is exactly why enzymes are so incredibly important in medicine and health.
Believe it or not, sometimes we actually need to stop an enzyme from doing its job. This is called inhibition, and it's the secret behind how a ton of modern medicines work. There are basically two ways to throw a wrench in the works. First, you have competitive inhibition. This is like sticking a fake key into the keyhole, the active site, so the real key can't get in. Or there's non-competitive inhibition. This is more like jamming the lock from the side, changing its shape so the real key won't fit anymore.
Let's make this real. A ton of people take medication for high blood pressure, right? Well, a very common type is called an ACE inhibitor. And here's how it works. There's an enzyme called ACE that normally creates a chemical that squeezes your blood vessels tight. The ACE inhibitor drug is basically that fake key. It gets in there and competitively blocks the enzyme's active site. So the enzyme can't do its job, that vessel-squeezing chemical never gets made, your blood vessels chill out and relax, and your blood pressure goes down. It's a brilliant way of hacking the body's own machinery to make you healthier.
Enzymes are also like little informants for doctors. See, many enzymes are supposed to stay tucked away inside specific cells in your organs. But if those cells get damaged, say in your liver or your heart, the enzymes leak out into your blood. So when a doctor runs a blood test, they can look for these telltale signs. If they see high levels of a liver enzyme like ALT, that's a huge clue that there's liver damage. High levels of a heart enzyme like CPKMB, that points straight to a heart attack. It's like forensic science for your body.
So, there you have it. Enzymes are nature's perfect little machines. They are absolutely essential for life, they're key to our health, and they're a cornerstone of modern medicine. Which, of course, leaves us with a pretty mind-bending question for the future. We've talked about them as nature's machinists, but what happens when we become the machinists? What could we achieve if we learned to design and build our own enzymes from scratch? What diseases could we cure? What problems could we solve? The possibilities are just incredible to think about.